Hello everyone, welcome back to our molecular biology series. Today we are going on a scientific detective story. How did scientists figure out that DNA is the genetic material? For decades, people didn't know. They knew chromosomes carried heredity, but what molecule actually stored genetic information? Was it protein? Was it DNA? Nobody knew for sure. It took multiple experiments by Griffith, Avery, and Hershey Chase to solve this mystery. And at the end, we will discover something surprising. DNA wasn't even the first genetic material. So let's dive in. By the 1920s, scientists knew that heredity or genetic information was carried on chromosomes. Mendel had shown inheritance patterns. Morgan had proven genes are on the chromosomes. But here is the problem: chromosomes contain both proteins and DNA. So, which molecule is actually the genetic material? Most scientists thought it had to be protein. Why? Because proteins are complex. Made from 20 amino acids, they can have endless variety. DNA seemed too simple, just four bases. So, how could something so boring store all the information needed to build an organism? The debate was on: proteins versus DNA. Who would win? In 1928, Frederick Griffith was investigating Streptococcus pneumoniae, a bacteria that causes pneumonia. He had two types: the S strain, a deadly killer protected by a shiny polysaccharide coat, and the R strain, which was rough looking, harmless, and without any polysaccharide coat. The S strain killed mice, the R strain did not. Even after Griffith heat killed the S strain, the mice survived. But then he tried a simple mashup. He mixed the dead S strain with the live R strain. The result? The mice died. Inside them, Griffith found live and shielded S strain bacteria. And the shield was the polysaccharide coat, which was initially present in the S strain but not in the R strain. The harmless R strain had picked up a transforming principle from the dead remains, stealing the instructions to grow the lethal polysaccharide code. Griffith had discovered that the blueprints for life could be passed from the dead to the living. For over a decade, Oswald Avery, Colin MacLeod, and Maclean McCarthy played detectives to unmask Griffith's transforming principle. They isolated the dead S bacteria's all the ingredients, proteins, RNA, and DNA, to see which one held the power to reprogram the harmless R strain. They used a process of elimination strategy with specialized enzymes. They used proteases that killed protein, but transformation still happened. Then they used RNAs that killed RNA, but transformation still happened. But when they used DNAs that killed DNA, everything stopped. Without DNA, the harmless bacteria couldn't transform. The verdict was clear: DNA is the genetic material. Even so, many scientists at the time were skeptical. It would take one more famous experiment to finally silence the doubters. The absolute certain proof came from Alfred Hershey and Martha Chase in 1952. They worked with bacteriophages, a special viruses that only infect bacteria, or they infect bacteria. A bacteriophage is simple: a protein coat surrounding DNA inside. When a phage infects a bacterium, it attaches to the surface of the bacteria and injects its genetic material inside the bacterial cell. The bacterial cell then makes more viruses. The question is, what does the phage inject? Protein or DNA? Hershey and Chase used a clever trick with radioactivity to solve the mystery of whether genes were made of proteins or DNA. They knew DNA contains phosphorus but no sulfur, while protein contained sulfur but no phosphorus. So they grew viruses or bacteriophages in two separate batches, one with radioactive phosphorus to tag the DNA and another with radioactive sulfur to tag the protein codes. Then they let these radioactive phages infect bacteria, and after that they used a blender to shake off the empty viral shells. The result, only the radioactive phosphorus was found inside the bacteria. Since only the DNA entered the cells to reprogram them, it proved once and for all that DNA is the genetic material. The Hershey Chase experiment confirmed that DNA, not protein, is the heredity material because only the radioactively labeled DNA entered the bacterial cells during infection. Let's try to draw and understand this. Before understanding Martha and Chase experiments, let's take a look at bacteriophage structure. So I'm drawing a simple phage just to get a clear understanding of what it is and how it works. So here is a bacteriophage. It looks like a lunar lander. This is protein coat outside and its DNA is packed inside its head, inside the protein coat. So this is the DNA packed inside the coat. And these are the tail fibers that help a bacteriophage in attaching a bacterial cell. So suppose this is a bacterial cell. So when a bacteriophage attacks a bacteria or it infects a bacteria, it uses its tail fibers and gets attached to the bacterial cell. So these tail fiber help in initiate infection by attaching themselves into the bacterial cell surface. Now that we know how bacteriophage looks like, let's get into Martha and Chase experiment. They did two experiments side by side. In one, they grew phages. This is bacteriophage. So in the first experiment, they grew phages using radioactive phosphorus or P32. If you remember from our DNA structure video, phosphorus is an important component of DNA. It is a part of DNA backbone. So in this experiment where they used P32, it makes the phage DNA radioactive because DNA gets labeled with P32. That means it becomes radioactive. P32. That means the DNA here is radioactive. But the protein coat remains non-radioactive. Because phosphorus is found in DNA but not in protein. In the second experiment, they used radioactive sulfur 35. So they grew some bacteria in this radioactive sulfur and that made their protein coat radioactive. because sulfur is a part of proteins because it is found in many amino acids. So these bacteriophages became radioactive, and I am using this blue color to show their radioactive protein coat. So coat labeled with S35, but their DNA remains non-radioactive. And why? Because sulfur is found in protein but not in DNA. So now you know the difference. Here at the left hand side, the DNA is radioactive, and here in the right hand side, protein coat is radioactive. In the next step, they let these labeled phages infect bacteria. So in the next step, these radioactive bacteriophages started infecting bacterial cells. So suppose this is a bacterial cell, and here this is a bacterial cell. After the infection started, they did something clever. They put everything in a blender, and this violent agitation knocked off the empty viral coats from the bacterial surface. Then they spun it in a centrifuge. So why they put everything in a blender? Because they wanted to get rid of these protein coats, radioactive or non-radioactive. So after the centrifugation, suppose this is after centrifuge. After centrifuge, what they saw was the bacterial pellet. This bacterial pellet was heavy, so it was found in the bottom of the tube, but the supernatant was light. So it was, this is viral coat, floating as a liquid as a supernatant in the tube. So when they examined this, they found that in this P32 experiment, they found the bacterial pellet was radioactive. But the viral coat was non-radioactive. They found radioactivity in the bacterial pellet. That means the DNA had entered the bacteria. The empty coats floating in the supernatant were not radioactive because the DNA had left them and gone inside the bacteria. In the S35 or sulfur experiment, they saw something opposite. So suppose here is our tube after centrifugation. So what they saw was this protein coat was this viral protein coat was radioactive. But this bacterial pellet was non-radioactive. Radioactivity was found in the supernatant with the empty coats. The protein never entered in bacteria. It stayed outside. The bacteria were not radioactive here. So If you think about it, bacteria which were infected with viruses or bacteriophages that had radioactive DNA were radioactive, indicating that DNA was the material that passed from the viruses to the bacteria. On the other hand, bacteria that were infected from bacteriophages that had radioactive proteins were not radioactive at all. This indicates that proteins did not enter the bacteria from viruses. So, therefore, the conclusion is crystal clear. DNA enters bacteria and directs them to make more viruses, but protein doesn't enter. It's just a protective coat. DNA is the genetic material. So, this experiment finally settled the debate. DNA, not protein, carries the genetic information. Now that we know DNA stores and passes on the heredity information, let's ask, what makes a good genetic material? A genetic material needs four properties. First, it must be able to replicate. That means make copies of itself. Both DNA and RNA can do this through base pairing and complementarity. Second, it must be stable, not changing with different life stages or conditions. DNA is chemically less reactive and structurally more stable than RNA. RNA has a reactive 2' hydroxyl group on every nucleotide that makes it easily degradable. RNA can even act as a catalyst. Think of ribozymes, which makes it chemically reactive. DNA is more stable. The third property is it must allow mutations for evolutions. Both RNA and DNA can mutate, but RNA being unstable mutates faster. That is why RNA viruses evolve so quickly. Fourth, it must express genetic information as proteins. RNA can directly code for proteins. DNA depends on RNA for this. So both can work, but DNA's stability makes it better for long term storage of genetic information. So, why is DNA the predominant genetic material while RNA does other jobs? The answer is DNA is more stable. It doesn't have that reactive 2' hydroxyl group and it uses thymine instead of uracil, which gives additional stability. You will learn why in higher classes when you study DNA repair. DNA is double stranded, so if one strand gets damaged, the complementary strand serves as a backup for repair. All these features make DNA perfect for storing genetic information long term. RNA, being more versatile and catalytic, evolved to handle dynamic functions such as carrying messages as mRNA, adapting as tRNA, and even catalyzing reactions. Different molecules for different jobs. Now, here is something interesting. Some viruses use RNA as their genetic material. Examples: tobacco mosaic virus, QB bacteriophage, influenza, HIV, and even COVID 19. All RNA viruses. Why does RNA work for them? Viruses have short lifespans and small genomes. They don't need long term stability. In fact, RNA's instability is an advantage. They mutate rapidly and evolve fast, which helps them evade immune systems and adapt to new hosts. That is why flu vaccines need updating every year because the viruses keep changing. So, RNA can function as genetic material, but for most organisms that need stability, DNA is preferred. This leads to a fascinating question: which came first, DNA or RNA? The answer is RNA was the first genetic material. There is strong evidence for this. Early in Earth's history, there was an RNA world where RNA did everything. It stored genetic information and catalyzed chemical reactions, both jobs. Even today, we see remnants of this RNA world. Ribosomes, the protein making machines, use ribosomal RNA to catalyze peptide bond formation, not any protein enzymes. RNA splicing is done by RNA molecules. Many essential processes evolved around RNA, but RNA being catalytic is also reactive and unstable. So, over time, DNA evolved from RNA through chemical modifications that made it more stable. And examples of uh, b- gaining stability were losing the 2' hydroxyl group using thymine instead of uracil and becoming double stranded. DNA gradually took over the job of storing genetic information. Proteins, being better catalysts, took over most enzymatic functions, and RNA remained as the messenger between DNA and proteins, and as adapter molecules like tRNA. Each molecule evolved to do what it does best. DNA stores, RNA transmits, and proteins work. Let's summarize this amazing scientific journey. Griffith in 1928 discovered something could transform bacteria but didn't know what. Avery and colleagues identified it as DNA in 1944. Hershey and Chase gave final proof in 1952. DNA has the perfect properties for genetic material, stable yet capable of replication, mutation, and expression. DNA is more stable than RNA, making it better for long term storage. Some viruses use RNA because they benefit from rapid mutation. And fascinatingly, RNA was actually the first genetic material in Earth's history, the RNA world, before DNA evolved as a more stable storage molecule. From mystery to answer, this is how science progresses, one brilliant experiment at a time. For NEET, please remember all these points. These experiments and concepts are frequently tested. If this video helped you understand DNA as the genetic material and RNA world, check out our complete molecular biology playlist. Drop any questions in the comments and I'll reply to all of them.